A very good evening, everyone. A warm welcome to all participants joining us today from across the country and the globe. On behalf of the biotechnology students at the Center for Scientific Research and Development, People's University, Bhopal, India, I'm delighted to welcome you all to the 10th session of the BioConnect webinar series, a special initiative driven by the student for the student. I'm Alicia Mansouri, and I'm proud to tell you that I am a PhD Biotechnology student and a member of the organizing committee for this webinar. It is a genuine honor to serve as your MC for today's session. Our session today focuses on a topic of growing importance in our field, dual use research and biosecurity, building a culture of awareness and responsibility. This webinar explores how we ensure that powerful scientific knowledge, which can be used to develop medicines and save lives, is always used safely and ethically. This responsible mindset is very essential for every biotechnologist. We are deeply honored by the presence of our respected dignitaries, our distinguished speakers, esteemed participants, faculty members, and our dedicated organizing committee members. I would like to extend a special welcome to our Honorable Registrar of People's University and our esteemed Professor Dr. Anthony Schwartz, Director of Biological Safety at Duke University, USA. Esteemed Professor Dr. Subhash Bhore, Head of the Center for Scientific Research and Development, and our advisor and mentor. To formally initiate the 10th session of the BioConnect webinar series, to invite our Honorable Registrar of People's University to deliver the opening remarks. Ma'am, please proceed.
Thank you, Alicia. Good evening, everyone. A very warm welcome to all distinguished participants joining us from India and other countries. It gives me great pleasure to welcome you all to the 10th edition of the BioConnect webinar series, a remarkable student led initiative organized by Center for Scientific Research and Development, People's University, Bhopal, India. This series truly embodies the spirit of for the students and by the students. It reflects the curiosity, leadership, and dedication of our young minds, who are not only learners but also contributors to the global scientific community. Today's theme, dual use research and biosecurity, building a culture of responsibility, is both timely and profoundly significant. In an era of rapid advancement of biotechnology, it is crucial that our research is guided not only by innovation but also by ethical responsibility and awareness of potential misuse. By discussing the concept of dual use research, we remind ourselves that scientific discoveries, while powerful tools for progress, must always be pursued with a deep sense of accountability, safety, and integrity. We are truly honored to have with us Dr. Anthony Schwartz, an internationally respected expert and director of the Biological Safety Division at Duke University, USA. Welcome, sir. His insights will undoubtedly deepen our understanding of biosecurity and inspire us to think critically about our roles as responsible scientists and researchers. I take this opportunity to commend the student organizing team from CSRD for their initiative, professionalism, and commitment in bringing together this global dialogue. Your efforts showcase the academic vibrancy and forward looking vision of People's University. I also acknowledge the guidance of Dr. Professor Subhash Bore, head of the Center for Scientific Research and Development, whose mentorship and vision have helped nurture this platform for academic and social engagement. To all participants, I encourage you to listen, engage, and reflect on how each of us can contribute to a culture of responsibility in science. Together, let us strengthen our collective resolve to ensure that biotechnology continues to serve humanity safely and ethically. Once again, thank you for being here, and I wish this session a great success. Now I hand over to our MC, Alicia. Over to you. Thank you once again. All the best.
Thank you. Thank you so much, ma'am, for those inspiring words. We truly appreciate your consistent support for student led initiatives at CSRD People's University. Now, I invite the chairperson of our organizing committee, Ms. Aisha Zamir, to share a few words with us. Ms. Aisha, over to you.
Thank you, Alicia. Respected dignitaries, esteemed guests, respected faculty members, and dear fellow students. Good evening. My name is Aisha, and I'm proud to tell you that I am an MSc Biotechnology student and a member of the organizing committee for this webinar. It is indeed a true honor to serve as the chairperson of the organizing committee. On behalf of the entire team, I extend a warm and heartfelt welcome to each one of you to this highly significant and timely webinar titled Dual Use Research and Biosecurity, Building a Culture of Responsibility. As we all know, modern scientific research has the power to transform societies and improve lives. Yet, this very power also comes with an equally great responsibility. In the fields of biotechnology, microbiology, and life sciences, discoveries intended for progress and human welfare can sometimes be misapplied in ways that pose risks to health, security, and the environment. This is what we refer to this is what we refer to as dual use research. In recent years, biosecurity has emerged as a global priority, ensuring that the benefits of research are maximized while the risks of misuse are minimized. The challenge before us is not only scientific but also ethical. How do we balance the pursuit of knowledge with the responsibility to safeguard humanity? What makes this discussion particularly vital is that biosecurity is not confined to laboratories alone. It calls for awareness, strong policies, and above all, a culture of responsibility shared by researchers, institutions, governments, and society at large. Only through such a collective approach we can prevent potential threats while encouraging innovation. To my fellow students, I want to underline your role with great emphasis. You are the future custodians of science. Be mindful of the power of knowledge and let integrity and responsibility guide your curiosity. As you explore and innovate, always remember the impact of your work extends far beyond the laboratory. With these thoughts, I once again extend my warm welcome to all participants. I'm confident that today's session will be a thought provoking exchange of ideas that will inspire us to reflect, question, and act responsibly in our scientific pursuits. Let us work together to develop solutions that not only advance science but also serve the planet. I look forward to an inspiring session filled with innovative ideas and meaningful discussions. Thank you. Alicia, over to you.
Thank you so much, Ms. Aisha, for your welcoming remarks. Distinguished participants and fellow students, to guide us through our speaker's presentation and manage the interactive question and answer session, I now hand over the session to our moderator, Ms. Forain Fatima Naqvi. Ms. Fatima, over to you.
Thank you, Ms. Alicia. Good evening, everyone. I am Conan Fatima Naqvi, and I'm proud to tell you that I am a first year MSc Biotechnology student and a member of this webinar's organizing committee here at People's University. It is truly an honor to serve as the moderator for today's enlightening webinar. We are honored to have with us Dr. Anthony Schwartz, who currently serves as the Director of the Biological Safety Division at the Occupational and Environmental Safety Office, Duke University, Durham, North Carolina, USA. Dr. Schwartz brings extensive expertise in biosafety, biosecurity, and the oversight of dual-use research. At Duke University, he serves in multiple key roles: biosafety officer, responsible official for the Select Agent Program, and institutional contact for dual-use research. His leadership extends across both research and clinical enterprises, including the Regional Biocontainment Laboratory. Before joining Duke, Dr. Schwartz made significant contributions at the National Institutes of Health, where he led the Select Agent Program after completing the NIH National Biosafety and Biocontainment Training Program. He's a PhD microbiologist and a certified biological safety professional with a career dedicated to ensuring safe and responsible science. Beyond his institutional roles, Dr. Shorts has been deeply engaged with APSA International and has advised on numerous global initiatives that build biosafety and biosecurity capacity. His international work spans India, West Africa, and other regions, focusing on strengthening oversight systems and developing skilled scientific workforces. Dr. Shorts is also a published author in the fields of biosafety governance, pandemic preparedness, and laboratory safety, and he's a frequent contributor to national and international forums that bridge scientific practice with policy development. With such an impressive background, we are truly privileged to learn from his insights today. His talk will shed light on how we, as researchers, educators, and professionals, can foster a culture of responsibility in the context of dual use research, a subject that carries profound implications for both science and society. Today, he will be sharing his insights on the topic dual use research and biosecurity, building a culture of responsibility. So, with this brief introduction, I now have the great pleasure of inviting Dr. Anthony Shorts to deliver his talk. Dr. Shorts, over to you.
Thank you very much. Everybody can hear me okay?
Yeah, we can hear you.
Okay, sounds good. So really appreciate the invitation to speak with you all today. And it's wonderful to see students being involved as much as you are and taking the time and initiative in addition to your studies to explore various topics and be knowledgeable about things that could impact your science. So it's a really privilege to speak with students. I remember, back to my undergraduate days of organizing things, being parts of associations, and it really is a happy feeling to see you being very active. And also, I really appreciate your leadership, Dr. Bore and Registrar, ma'am, for inviting me and the kind words as well. And hopefully, I hope this will be very kind of informal, because if you have any questions, I would like you to just stop me and ask. Also, I'm presenting from my laptop, so I cannot see the chat while I'm talking. So just feel free to unmute and ask a question if you'd like. This is one of many topics that I deal with at an institutional level. So there's other things like biosafety and all the day-to-day stuff that we deal with. So if you have any questions about that, I'm happy to get into that either now or later as well. So with that, one last thing I would like to say is I was born and brought up in Chennai till I was 15. Then we moved to the US. I'm sure you guys know where Chennai or Madras is. I still have ties to India, and it really matters to me that research is done safely there. And young people like you, when you're performing your research, my hope is that you pay attention to biosafety and biosecurity and do the research safely to protect yourself, your communities, so that you can do great things. Continue to work on great projects and do it in a safe manner so you can go on to make discoveries and cures and all those things. So again, happy to be here. And with that, I'll share my screen and get started. Sounds good? Yeah. Let's see. Okay. Hopefully everybody's able to see my slide deck. If not, let me know, but I'll continue as is. So you guys heard about all the different roles that I play, so I'm not going to get into that right now. But if you have specific questions about biosafety and biosafety as a career and what types of roles there might be within that and all the different hats that I wear at Duke University, please feel free to ask questions about that. I'm happy to explain to you what each of those mean. But I wanted to start off with a little bit background on Duke University and Duke University Health System. Um, you may know that we're one of the top ranked institutions quite recently, and this, of course, changes year to year. We were ranked number 6 in the United States in terms of education, research, and other things. We have anywhere approximately 6,500 to and then 10,000 or more students, professional students. Undergraduate students are around 6,500. In terms of research, sponsored research that we do, it's close to $700 million just from the NIH alone. If you, this is per year, so if you factor in other funding sources such as DoD or NSF or others, Duke typically has more than $1 billion in research funding. And in that, a subset of it is PIs, about 600 plus principal investigators, PIs, who have wet labs. So, wet lab research versus other things, theoretical research. So they have wet labs, and this is what I have oversight of when it comes to biological hazards in those wet labs. We're also part of the NIAID network of BSL-3 and BSL-4 labs. So Duke has a regional biocontainment laboratory, and again, I'm happy to share what that means, where we have biosafety level 3 and animal biosafety level 3 laboratories. We're also part of a large medical teaching hospital network. We have 3 different hospitals, 1,500 plus beds, and primary care, specialty care clinics, around 200 plus. And we see quite a bit of patients. As you can see from the slide, 5 million outpatient visits, more close to 67,000 inpatient stays. So a lot of interaction with patients and infectious diseases, and that we also have to protect our clinicians, our nurses from using biosafety methods. And then finally, we are a big institution, including our health system. There's 43, and now I think it's much larger, 47,000 employees. So when it comes to protecting them from biological hazards at the workplace, that responsibility falls on me and my team. So where do we fit within the overall picture? My division falls within what's called the Occupational and Environmental Safety Office. We, OESO, as other divisions, as you can see, biological safety is one of them, but we are part of an overall safety office that makes sure those 47,000 employees, students, everybody else is protected in their day-to-day activities on our facilities, right? So things like ergonomics or laboratory safety, fire and life safety, occupational hygiene. So for example, respirators, when people wear them, there's a lot of requirements. That's our occupational hygiene group takes care of that. Radiation safety. So if you're doing any research or patient care with radioisotopes, there's a whole lot of regulations that must be followed. So, these are things that people in my office, the OESO office, go out there and take care and make sure everybody's doing things safely. So my team, biological safety, as I mentioned, which I'll keep saying, is more focused on biological hazards. And in the laboratory environment, we tend to focus on more higher risk biological organisms and hazards from them. So this is my team. People with varying experiences, varying times at Duke, but they do the bulk of the work and make me look good. So I want to acknowledge my team and say that it takes a lot of people to get things done, especially when it comes to safety. So hopefully this also kind of gives idea of Indian institutions how just this is my division, but all the other divisions that you saw earlier, we have about 65, 70 employees taking care of safety issues at the institution. So biological safety, where do we fit in and what all do we do? Our role spans a spectrum of things here at Duke. anywhere from environment of care. What that means is when you walk into a hospital or a clinic, we make sure that certain safety measures are taken care of, which means my staff go out there and do audits and look for things that could be improved. And then we work with the nursing as well as physician groups to try and make sure that everything is safe for the patient coming in. We also have oversight over clinical research. So whether that's reviewing the research and approving the safety from an employee handling perspective, we do that. We don't get into what it does to the patient. We have what's called the Institutional Review Board, IRB, that deals with the patient side of things, but the employees handling those clinical material, we have oversight of that. Next is clinical laboratories. So let's say you go to the doctor and they have to take some blood cultures or urine samples, whatever the case may be. They get sent to different laboratories, right? Diagnostic laboratories. So just like that, Duke has many clinical laboratories that deal with patient samples, potential infectious agents. So we work with them to make sure that they're doing all of these safely. Part of that is also our autopsy and pathology labs, and you can understand kind of the risks from sharp devices when they're doing autopsy, whether that's to figure out what sort of diseases the person had or some other reason. So we have to protect our employees from those type of hazards. Research is the bulk of things that we deal with, whether that's a biosafety level 2 or biosafety level 3 lab, select agent program, and just regular laboratory work. So we work a lot with lab personnel, PIs, as well as the laboratory workers, students, to make sure they do the work safely. We also have a role in animal research. So as you know, animals are used in research as model organisms, and they could be infected with some pathogens that then we have to make sure that in the process of putting it into the animal, staff don't expose themselves to them. So going into the animal, it could be different ways, right? You can inject it, you can aerosol exposure animals to pathogens, for example. Then we have to make sure what safety precautions are in place to ensure that staff themselves don't get sick from it. And then last but not least, definitely our facilities folks who make sure that all of the locations, the laboratories, hospital environment, all of these are functioning well. We make sure we work with them to protect them when they need to go into these spaces and do the work that they need to keep us functioning. So as you can see, it's a wide range of individuals we deal with, locations we deal with. So it keeps the job very interesting. So if you ever are thinking about going into the safety field, one of the things that we like to say is that no one day is the same. So it really is interesting because you're dealing with so many different things. And also you get exposure to various areas of science. It's not one very narrow focused, your subject. It's various topics that you have to learn about. You have to learn about the hazards that they present and what kind of experiments the laboratories are doing, and do a lot of risk assessment, and it keeps things very interesting. So in the laboratory environment, I won't go into all the details here, but in the laboratory, when a new lab is set up, we consult with the laboratory and along with our lab safety division and make sure that they have all the proper safety measures in place. At that time, we try to understand what it is that they work with and how they're planning to do the work, so that we can put all the safety measures and tell them what training they need to take and things like that. We also conduct audits, which means we have to make sure if we're telling them these are the things you have to do safely, we have to make sure that they are actually doing it. So we visit them at least annually and their lab, look at everything that's in the laboratory and their work practices, and tell them, if something needs to be fixed. As I mentioned earlier, we do have a regional biocontainment laboratory, which is a BSL-3 level laboratory. And then also we have two other BSL-3 laboratory spaces. So for that, there's a lot of requirements, technical requirements. If you're not familiar with the BSL-3 laboratory, one of the important things to remember is it has a negative airflow, meaning you try to keep all the pathogens inside. So any aerosols generated, anything else, you try to keep it inside by pulling air into the laboratory in a very simple form. Of course, there's other personal protective equipment like PPE, where you can wear respirators. There's a lot of procedures that you have to follow. But at the very minimum, if you need to kind of remember how BSL-3s are different from, say, a regular BSL-2 laboratory, you're pulling air into the laboratory and keeping it negative to the outside. Therefore, all the pathogens can stay inside if there's any sort of aerosol. Typically, there shouldn't be anything because you're working at a biosafety cabinet, but this is an added measure and an added precaution, so you can continue to research those organisms and do it safely. So there's a lot of requirements associated with that. A subset of those pathogens, it could be in BSL-2 or BSL-3 or BSL-4. Those are the levels. There are 4 levels, biosafety levels, 1, 2, 3, and 4. 4 being the highest, where you work with really dangerous pathogens like Ebola, Marburg virus, and things like that. 3 is where you work with things like TB, monkeypox, those type of things. 2 is more your standard things that most people shouldn't get sick from. So Staph aureus, Pseudomonas, all these things you work with level 2. And then BSL-1 is more your microbiology laboratory in a college environment where it's really safe to work with those things. So a subset of agents are part of what's called a select agent program. What that is a list of about 65 to 70 viruses, fungi, bacteria, toxins that are on this list by the federal government. And in order to work with that, you have to go through a security clearance process. You have to have additional training. Depending on certain agents, you have to go through a suitability program, which means we have to make sure that you're trustworthy to work with them. We have to conduct drills. We have to look at your inventory and make sure that everything is proper. There's no theft, loss, or release of your samples. And also we get inspected by CDC and APHIS, which is part of our U.S. Department of Agriculture. So we get inspected, then if they find issues, we have to fix them, but there's a lot of regulatory aspects involved with that. And that's my role as the responsible official of the Select Agent program for Duke University to make sure we are in compliance of that. And what I'm going to talk to you today is about the dual use research of concern and pathogens with enhanced pandemic potential policy. So what this, and I'll go into a little bit detail, but what this is about is any sort of work that has a potential to be used both for good and bad purposes. So really depends on the intent of the individual. So what we try to do is there's a program, which I'll go through the background of how it all originated. There's a program that then we have to administer, make sure our staff are complying while they're doing all this important research. So as I mentioned earlier, this is one kind of subset of the various things we deal with that I just showed to you. So if you're interested in other things, I'm happy to discuss, but today's topic is dual use research. So what is dual use research? Just to make sure that we are all on the same page, here's some basic terminology. Life sciences research that could be misapplied to pose a significant threat to public health, agriculture, or national security. So dual use research of concern is basically that if life sciences research that you're working on, thinking in the back of your mind that I'm working on a cure, I'm going to find, how this pathogen works, all of that, then somebody could take that knowledge and misapply that to cause a threat to public health, agriculture, and national security. The other terminology that gets used in this realm is PPP, which is pathogens with pandemic potential. So these are things, as you know, with COVID, we recently experienced this. SARS-CoV-2, that's a coronavirus that had the potential to spread throughout the world, right? So similarly, influenza virus, SARS, the first SARS that came about in 2013 or so. So these all have the potential to just kind of explode and go into the entire world and cause a pandemic, right? So that's PPP. So you will also hear terminology EPPP, which is enhanced potential pandemic pathogens. And this is typically found in one of the policies, but no longer used. is basically any alterations that you make to these pandemic potential pathogens, then in terms of transmissibility or virulence, because you want to study that, then that kind of becomes the enhanced pathogens, enhanced PPP. There's another framework that is now all kind of bundled together. It was called the P3CO framework. Basically, what it was is these pandemic pathogens. There was some oversight of mechanism for how people should work with it, what sort of reviews needed to happen. So that was the P3CO framework. The other kind of terminology, which is much newer, is called pathogens with enhanced pandemic potential, similar to the PPP that we, or EPPP that we talked about earlier, but just more of a newer, more clear terminology. And then one that you probably have heard quite a bit of is gain of function research. Basically what this is any sort of modifications that you might make, and very simply to a pathogen that could potentially increase its transmissibility, host range, virulence. That's called kind of gain of function research. One of the drawbacks of using this terminology, gain of function research, is many times in the laboratory, you might do gain of function research to study very benign things, right? You might modify a pathogen to see if it is able to, for example, use a different nutrient source, right? So yes, it could gain that function, but is that really a bad thing? Is that in the grand scheme of things, in terms of research that could cause harm, is that gain of function research? So there's a lot of debate about this terminology. So just remember that not all gain of function research is dual use research or PP research, right? So I just wanted to put this out there, these terminologies, because you will hear me use it going forward. So didn't want you to be any confusion, but if you have questions, feel free to ask. So why is this important? Throughout history, biological agents have been used as weapons, and this is even before recent war times. For example, in World War II, both sides, Allied and Axis powers, they used or explored the usage of biological weapons. Japan, for example, had this unit, military unit called 731, who conducted experiments on humans using Yersinia pestis, which causes plague. And they did this in one area of China, which they were occupying. United Kingdom, for example, planned to disseminate and certain anthrax spores and tested in a particular island off the coast of Scotland. US, for example, started a biological offensive program in 1943 in this place called Fort Detrick in a state called Maryland, and then which later was disbanded. But many countries have been exploring this, and even if you look back in like really old, back in the days, there are lots of examples of people contaminating water supplies. They didn't know maybe how it was happening, but contaminating water supplies or throwing dead bodies of infectious people who have died in infectious disease across like a fort, for example, so that people inside would die and then they can take over. So people have been doing these types of things and using biological agents to cause harm country to country. Then also biological agents have been used by bioterrorism as a bioterrorism weapon by different groups. For example, there was a Rajneesh movement in 1984. This group was you can look this up, it's on Wikipedia. This is an Indian kind of holy man who gained a following and then moved to the US. They were trying to take over a particular city and to influence kind of the local elections that were deciding whether to kick them out or keep them there. These people ended up poisoning salad bars at the various restaurants with Salmonella enterica typhimurium to poison people so they wouldn't go and vote in those elections. There's also a Japanese cult, Aum Shinriko, in 1993. They sprayed Bacillus anthracis spores from a cooling tower that was on top of their headquarters. Thankfully, it turned out to be a vaccine strain that didn't really cause issues. And of course, individual actors have used biological agents as well. For example, right after 9/11, when the plane crashed into the Twin Towers, that happened in September, and then in October we started seeing different places receiving anthrax spores, for example, in the mail. And when they reached certain locations, people were exposed to this, and there were 5 deaths and 17 people were sick. And it caused one of the kind of biggest law enforcement cases in the US. FBI did a lot of follow-up. As you can see, about 10,000 interviews, 6,000 items of evidence. And charge was brought against a scientist in one of the US Army laboratories. But before they could send him to trial and whatnot, he killed himself, and it was never really fully proved that he was the person behind it. But the reason I went through this is you can see that at various levels, whether the state actors or groups or individual, and there's plenty more examples, people have tried to use biological agents to cause harm to others. So just as off the bat, just one of the things as students and people working in the laboratory, things to remember is you can keep those agents that you're working with safe, right? It could be a pathogen, but maybe not a pathogen, but still at the end of the day, you can follow some security measures to keep these things safe, because somebody with bad intent shouldn't use that for ill purpose. So that's something to just a takeaway. But what happened was, just going back to this, is right after 9/11 happened, there was a committee that was formed, and in 2002, that's a typo there, 2002, right after 2001, 2002, a committee was formed to look into how biological agents could be misused. So, or biological technology, biotechnology could be misused. And this report was called Biotechnology Research in an Age of Terrorism. The person who led that, the chairperson, is Gerald Fink. So just over time, this report started getting referred to as the Fink Report. And if you Google Fink Report, you will see this report if you would like to read it further. But what this report did was, it's a comprehensive study, one of the first comprehensive studies to look at dual-use research of concern in biotechnology. And what it tried to do was balance scientific openness with national security. So as you mentioned, your students very eloquently put it, it's important to do the scientific research, but at the same time, you have to do it safely and securely. So that's really what the balance they were trying to strike in this report that came out more than 20 years ago. And we're still kind of grappling with this issue. So one of the things that they did was define this concept of experiments of concern that could be misused for bioterrorism. And I'll show that slide in a second, but other recommendations that came out of this Fink report was to say, foster a culture of responsibility among life scientists, which is all of you who are working in the laboratory. So everybody needs to take responsibility to do things safely and securely. They also recommended that the US establish a national advisory board. Basically, it's called the National Science Advisory Board for Biosecurity. They talked about education and training to incorporate dual-use research training, biosecurity training into scientific curriculum. Many places are starting to do that now, like there might be a one-semester course, or there might be a guest lecturer who would come and talk to people about dual use and biosecurity. And also, the other things that they talked about is get journals and editors to look for risks of publishing this type of information. Even if, say, for some reason, the institutional oversight was missed and you're submitting a publication your paper to be published, they wanted the journals also to have some mechanism in place to say, okay, could this be dual use? What if this information gets in the wrong hands? There should be some dialogue about it, some oversight, and make sure that they are able to do that review. And also promote global dialogue. As we saw with COVID, infectious disease don't stay in one country, right? Even if they originate there, they don't just stay in one country. There's no borders because people move from country to country, diseases spread. So whatever happens, say in India, can impact us. What happens here can impact in India and all over the world for that matter. So it's really important to create that awareness with everybody and then get everybody to kind of start thinking about these things. And of course, regularly monitor biotechnology advances for any new risks. I'm sure you've all heard about synthetic cells being developed, gene drive technology, gene editing technology. Could any of those things be potentially dual use technology? Can that get into the wrong hands and somebody does bad with it? So those are things that is what they meant by let's look at as technology advances, which definitely will. How do we make sure that those are also assessed? And then at the institutional level, which I hope to kind of show you some examples of how we do it, so it can be also done in other places, is there's a review process, there's a institutional committee, and other trainings and things involved, and that's what the recommendations mention. So the 7 experiments of concerns, we kind of colloquially say 7 deadly sins, are experiments that would demonstrate how to render a vaccine ineffective. You can obviously see why that would be a problem. Somebody has a vaccine for a very deadly infectious disease. Now, if you showed an experiment how that could be negated, then that's an issue. Would confer resistance to therapeutically useful antibiotics or antiviral agents. So again, if there was a antibiotic that you can take to cure a bacterial infection, now you're showing experimentally how to overcome that, that's an issue. Would enhance the virulence of a pathogen or render non-pathogen virulent. Again, we are already struggling with controlling a pathogen. If you do any experiments that would make it much more virulent and you're putting that information out there, then that's an issue. Increase the transmissibility of a pathogen. So this usually comes in place with the pandemic potential pathogens that I mentioned earlier, like flu, SARS, SARS-CoV-2. These are things that just naturally spread, but if you modify them to make it more transmissible, then that's obviously an issue that needs to be reviewed. Also, alter the host range. Let's say there's a pathogen that now goes from fruit bats to pigs, but that's kind of the dead-end host. But you are worried that someday it could jump to humans. So now you're starting experiments to review that or to go in that direction and see what would happen, what would take for it to jump to humans. That experiment, when it's not in nature and you're altering the pathogen to cause the host range to change, that is something that you definitely need to review and make sure is done safely. And then also any sort of evasion of diagnostic or detection modalities, that's important for to make sure that those type of research is reviewed. And last but not least, the Fink report said anything that would enable weaponization of a biological agent or toxin. You can see, except maybe for the last one, there's some benefit to trying this with good faith and to see like what would happen if this naturally happens. Can we get ahead of that and do some of this research so we can be ready when naturally pathogens evolve? But somebody can take that good research that you're doing and use it for bad purpose. So that's why these are all problematic. So as a result of it, here in the US, the Fink, based on the Fink report, there was an advisory board set up, NSABB, and also these 7 categories became kind of the foundation of defining dual-use research. There was concerns raised globally at the Biological Weapons Convention, and publication of sensitive data was debated, and there's a good example of that. There's also what this did was bring scientific self-governance and government oversight into kind of one big ball, and people are still trying to figure out how to do that best, and also laid the groundwork for many policies that came from that. I'm going to speed up a little bit due to time, but basically in 2012, U.S. government put out what's called federal level policy on DIRC. Basically, in that policy was aimed to funding agencies, because a funding agency like say NIH or DOD if they're going to fund a research, they want to be careful that this doesn't inadvertently have dual use potential. So this policy was aimed at that, so the funding agencies can have a review process and make sure they're not funding dual use research. In that, how they define dual use research was that based on current understanding, can be reasonably anticipated to provide knowledge, information, products, or technologies that could be directly misapplied to pose a significant threat with broad potential consequences to public health, safety, agriculture, crops, other plants, animals, the environment, material, or national security. Here, material is about things that can be used for defense purposes. So you can see how it is broad, but at the same time, they try to narrow it and give a very kind of concise definition on what should be concerning. But as people started reviewing this type of research, it's really not that easy. It's a very subjective thing because many places do this kind of research. Some information is already out there, and also research evolves. Like when you start a research project, it may not be obvious, but as you're going into the research project, you have to keep in the back of your mind, like, is the result from what I'm doing, could it be dual use? So those are very tricky things to review and come to consensus on. So an example, for example, if you mutate a gene or many genes in a pathogen to study drug resistance, transmissibility, or host range, and so that you can come up with drugs and be ready when it happens in nature, somebody can take that information and misapply it, right? They don't care about the good that would come from it. They care about causing harm with it. So basically, in a very short way, good science that can be put to bad use by bad actors. So that particular policy had these 15 agents that it was concerned about because they thought these are really bad ones. And then they also defined had their own definition in the policy of those experiments that shouldn't be done. I'm not going to go over this fully, but basically it's kind of the same as we talked about before, but some rewording, some clarity, some trying to be concise and helpful. The one thing that I do want to point out is G, the last one, which is a bit different than before. So what it says is any experiments that generate or reconstitutes an eradicated or extinct agent or toxin is problematic, and you can see why. For example, something that we haven't seen in hundreds of years, if you bring that pathogen back, let's say you found a sample in an archaeological dig or in the Antarctic or somewhere, and you bring that back, obviously the current population doesn't have immunity to that. So the concern is that it could spread and then become an issue, right? So just a quick example, this caused a lot of controversy. I'm going to go through it quickly, but if you have any questions, I'm happy to answer. There was in 2011 an experiment that was done with ferrets in influenza. The H5N1 strain of influenza was modified by these two professors, Ron Fushier and Yoshihiro Kawaka in these institutions. And they identified exactly which mutations would make it more transmissible and more fatal, in mammals. They used ferrets as their animal model. And this kind of information went into the publication stage, and that's where they said, wait a minute, this could be an issue. So again, the NSAB, which was established, they were called, they were debated it, and they said, let's not publish this. It has to be redacted, some of the methods. And that was kind of like the first call for censorship, which is, coming from a scientific community, I'm sure you all understand that if you spend hours and hours in the laboratory doing your research, you want the world to know about it. But to tell you that, no, you cannot publish this can cause a lot of issues. So the World Health Organization and many scientists pushed back, and the paper was finally published in full in 2012. This caused a lot of debate around gain-of-function research and dual-use research and how publications can be controlled. Then what scientists did, they voluntarily went into a moratorium for this particular kind of research. Initially, they said, let's 60 days, take a pause and look at it. But then it ended up turning into a year-long thing. Why? Because in 2014, gain-of-function research funding itself was paused because the federal government said that, we need to look into these things, especially research with influenza, SARS, MERS, all these other things. And they wanted to put a framework in place to do this kind of research. And then, National Science Advisory Board for Biosecurity, 2016, they proposed a framework. They said there needs to be a structured approach. If you're doing this kind of research, there has to be some risk-benefit analysis. The benefit has to outweigh the risk, and why there's need to do this kind of work. There needs to be oversight at different levels, and also good mitigation measures need to be in place. And then also in 2017, a new policy was put out, which is what I mentioned earlier. It's the Potential Pandemic Pathogen Care and Oversight Guidance, which talked about how to work with it and what kind of oversight is needed. So what happened in 2014, the funding agencies, when they received their policy in 2012, they pushed down a policy for the institutions to do this kind of work and have this oversight. So 2014, they said every institution that does this kind of work needs to have what's called an institutional review entity, and a person has to be assigned institutional contact for dual use research, ICDR. A person has to be assigned who can be the point of contact to deal with all these issues. And for Duke University, that's one of my other roles. And the PI and lab people who are working with these 15 agents that I showed earlier have to be trained and made aware that this is a policy and these are things you need to watch out for. And this policy also made it kind of a shared responsibility between researchers, institutional review entities, journal editors, publishers, and the federal government. So it became, more broader and trying to actually put a framework around things. And I want to just briefly mention that in 2024, last year, there was another policy update. Basically, it merged all those policies that I just talked about very briefly. There's lots of details in those policies, but merged it together and said, let's deal with it in one kind of structure. And what it also did in the process went from 15 agents to 90 agents, and for dual use research of concern. And they also broadened from those 7 outcomes to 9 outcomes and added a couple more. For PEPP research, there's no specific agents or lists because, things becoming potentially pandemic, depending on how you modify certain pathogens, anything can become potentially pandemic. But they also focused on 4 outcomes for these. What this policy also did was it made it a broader institutional response, meaning most U.S. organizations, U.S. institutions have a sponsored programs office, means these are the people who help manage all the grants that are coming in. So it kind of brought them in, brought leadership in and said, everybody has to deal with this issue. And the implementation deadline was May 6th of 2025 this year. So what we did, and again, I'm happy to talk about this in detail, but I'm sharing this so that there's some good ideas where people, if you want to do it in your institutions, you can put some of these things in place. What we did was, I won't go through this, I'll just show and tell. We mapped out who are all potentially involved and what are the different, stop and go places, where does the process end, who needs to notify all that. So we brought together, worked with a lot of people in different offices, brought together and mapped out this process. Then we developed a training for the PIs and grant managers who have to be part of this process. So now again, like I said, it's not just a safety office anymore. It becomes a broader kind of training and initiative. We also did a lot of communication, updated our website to include a complete section just for dirt pet policy, where people could go and look for information. We came up with a frequently asked questions section where they can find out what's going on. This is still on our website, so please take a look if you're interested. And then also in their grant tools, we started asking these questions saying, does your research fall under this? If they said yes, then we would ask them a more detailed question where they can go through this form that was built specifically for this, and they can answer some questions that would tell us what we need to do further. I know this is small, but just to say, like, what are our review process? What's the oversight process? when it goes to the committee, and all of those things are highlighted here. And we really had to, over a year's time, work to implement this. We also created risk-benefit analysis templates for our researchers. We created biosafety and biosecurity SOP templates for our researchers. So, when they do this work or when they want to do this work, they have to follow all of these things. We want, as much as we can, we try to facilitate the research. We will not say, full stop, you cannot do this. If there's a clear benefit in that research and it'll help people, we try to do that, help them do that as safely as possible. So that's why we put policies in place that they have to follow if they want to do this kind of research. But what happened, as I said earlier, May 6th of 2025, this policy was going to go into effect. May 5th of 2025, one day before, there was an executive order that was published by President Trump. And basically they said, we are concerned about dangerous gain-of-function research. Stop all the other things that you were doing. They had their own definition, own research kind of outcomes different from the 2024 policy, and then said, we will not fund that type of research, primarily foreign countries and here as well, and asked us to look at things at our institutions that fall under this newer, slightly different definition, and said that by September, so this came out in May, by September, they said a new policy would come out and will tell you what to do more. So September has come and gone. We haven't seen any new policies. What we have seen, though, is they've been trying to identify research that falls under this new definition so that they can address it. And some labs, again, because of time, I'm kind of speeding through this, but some labs have been asked to stop doing certain work. Their institutions, the principal investigator, they don't agree that this falls under dangerous gain-of-function research, but still the government has said, you cannot do these things. There's potentially other research that could fall under this, and more notices are coming out. And also, one of the things that happens, I'm not sure how this happens in India, but when new policies are put in place, typically what happens is there's kind of a proposed policy, then they ask for comment from the public, those who are impacted by it. They'll say, by this date, you have to send your comments and concerns, and we, the regulated community, would send our comments and concerns and say, this is not clear, or maybe do it this way, and they'll take that, and review it, and sometimes make changes, sometimes they won't make changes, but at least there's like a process by which you can try and do that. For this dangerous gain-of-function research, so far we have not had anything. So what we've had to do is kind of keep things in place for now, what we had, and internally kind of do some of these reviews. So that's where it stands. What I wanted to take last couple of minutes was to show that, how DIRT-PEP gain-of-function research kind of happens, or oversight happens in India. In the current framework, there are many biosafety related things. So the strengths, which are listed on the left, they're GMO, genetically modified organisms, review committees for genetic manipulation and biosafety, DBT, ICMR has several guidelines. A comprehensive guideline came out in 2017. It has all the information that you need to follow when working with biological agents, infectious agents, and also India participates in the Biological Weapons Convention and does all these things. But in terms of actual practice and actual implementation, there's no, when it comes to DIRT-PEP, there's no explicit dual-use research policy. There's no specific list or anything of things you cannot do. There's no national advisory board in this topic. There's no gain-of-function or PPP review process. And currently, the focus is on biosafety. And even that, there's not a lot of implementation that happens at the lab level. But biosecurity and dual use are really not discussed as much. So there's a lot needs to be done in terms of awareness. So as I was thinking about, what we have gone through, and, if India wants to put things in place in India, it would really benefit from learning from the things that have happened here in the US and proactively kind of build this oversight process and learn from some of the mistakes that have happened here, learn from definitions that we have tried to develop and things like that. What you get is the last 20 years of us trying to hash out and develop processes and make mistakes and do different things. And you can learn from that and start and put policies in place that make a lot more sense now after many things have gone through. And one of the things I would say is involve different stakeholders and consider varying viewpoints so that it can be evenly done, not just say government labs, for example. It include institutions, universities, anybody who's doing research, include them so that you can get different viewpoints. And also, India has an opportunity to lead the global south when it comes to kind of combining biosafety strength and developing these new awareness things. There's a lot of work happening in Africa in terms of biosafety. There's Southeast Asia as well, places like Malaysia, Thailand, different organizations work on biosafety issues. But in terms of DIRC, PEP, and biosecurity, a lot could be done, and India can really take the lead on it in the global south. And most importantly, and as I said in the beginning of this conversation, is students and early career researchers. They are going to be really key for keeping this issue in the back of their minds, and as they do their research, to start incorporating these safety processes and security processes, making sure that their research doesn't end up becoming used for bad purposes. And that kind of creating that cultural awareness is really important for the future. And India can definitely lead the way in that. So with that, I'm happy to take any questions.
Thank you so much, Dr. Shorts, for such a rich, insightful, and thought-provoking talk. Your talk has deepened our understanding of dual-use research and biosecurity. Building a culture of responsibility is a collective responsibility, we learned. Now, we will move into the question and answer session. I encourage all participants to keep your questions ready. You may type them into the chat box, and we will do our best to address as many as possible within the time we have. So I can see a question from Tanisha Soni. So may I read, Dr. Shorts?
Yeah, please go ahead.
Yeah. So, Dr. Shorts, what practical steps can young researchers take in their early careers to ensure they are conducting dual-use research responsibly while still promoting scientific innovation? This is the question.
That's a good question. I think one of the main things, as I mentioned at the very end, is awareness on what could potentially become dual-use research, right? Definitely educate yourself, learn more about what these research areas could be that could end up becoming dual-use research. And as I said, it's very subjective. It is even scientists who are in the field for a long time, regulators, biosafety professionals, biosecurity professionals disagree on a lot of things on what makes dual-use research. So I would say that it starts from kind of going out there and looking at what information is available, what is considered dual-use research, why people debate these things, and what are the potential issues that can come from this, and really start learning about that so that when you're doing your research, you can keep that in the back of your mind. And if anything comes up, you could explore it. If it's nothing, then you keep going. But if it is an issue, then it can be addressed with proper oversight.
Thank you, Dr. Shorts. Second question we got from Nandana Tiwari. So the question is, are there any specific national or international policies that regulate dual-use biological research?
Yeah, so as I mentioned, in the US, there are dual-use research policies and policies more at the policy level right now. There are laws trying to be passed. There are varying degrees in terms of how strict they are. There's one law, if you're interested in looking that up, called Risky Research Act that is going through. Again, there's a dangerous gain-of-function research policy that we're waiting to see when it'll come out. There are different things, right? And then it's a balance between the work that needs to be done to protect ourselves and come up with these cures and treatments, and how to do it safely. There will be groups that will say, no, you just don't do this work at all. And you are creating the issue. But there are groups that will say, like, no, this is naturally going to happen. We need to be ready for it. So it's really a balance. And as far as I know, there's nothing in India regarding policies or any regulations. In the US, there is, and we're kind of waiting to see what's coming up next. Yeah.
So thank you for that clear explanation. It really helps us grasp the essence of dual use concerns in biotechnology. So building on that, in your view, what are the biggest challenges in balancing scientific innovation with the responsibility to prevent misuse of biotechnology?
Yeah, in terms of biggest challenges, I would say is how do you convince different groups what the risk benefits are, right? Obviously, researchers feel very deeply about why they need to do this kind of work so they can be ready for whatever's coming up next, the next pandemic, the next outbreak. And they can see, and this has happened before in terms of influenza. People said we need to study influenza because there could be outbreaks in the future. And the US is currently dealing with an influenza outbreak in dairy cows and others. And there's concern that it could jump from species and become more of a widespread issue. So there is some solid reasoning and benefits in terms of studying these things. But then different groups are involved, and you have to be able to communicate your science clearly. You have to be able to communicate the benefits clearly and help each other understand why certain things can be done. The other thing I think also is people don't understand a lot about the laboratory procedures in place to do this kind of work safely. There's a lot of misconception on what biosafety means, or people don't even know what biosafety is. I mentioned earlier some of the procedures we have in place, from training people, PPE, engineering controls, building level controls. All of these things work together to enable researchers to do the work safely. So people don't understand that, and we need to do a better job of helping people understand it so that they can say that they can see that work can be done safely. Most things can be done safely, and there's always going to be some line that you draw and say this is something we cannot do. But everything else, I think, if you do it safely, you can follow the rules and do it. Things can be done safely.
Yeah, so Dr. Shorts, we are getting more questions from the audience. So the next question is from Tanisha Soni. It is a very interesting question. So the question is, how can India adapt successful elements from international dual-use research concerns oversight models, such as the US NSABB, while considering its own scientific and regulatory landscape?
Yeah, great question. India is a country that is trying to establish itself in various fields, right? It's not just biotechnology, automation, AI, quantum physics, all sorts of things. And that's important. As a nation, any country should aspire to do that. And it's also tied to a country's economy. You do better, innovate, then you would be able to provide for your people and do great things. So biotechnology is tied to bioeconomy. I'm sure you guys have heard of that. India has a big bioeconomy plan to use biotechnology to do great things for the population, increase GDP and things like that. So I would, first of all, help government regulators and others understand that by doing it safely, you can get to that point. By doing it safely and securely, you can actually make those things happen. If there's some mishap happening and it causes a pandemic or whatever, then you lose confidence in that process, in science itself, right? And that can impact, people can then get, the population can get pretty upset that they didn't do it safely. So first would be to kind of get that buy-in from leadership and make those things happen. And really think through what the oversight will be for that's good for India. And that doesn't stifle innovation, because there's always that push and pull between global north and global south in terms of, you've done your advancements, now it's our time to do the advancements, don't put these restrictions in place. It shouldn't be really seen like that, but it should be seen more like what works locally for India and how well we can kind of work within that framework, but still doing everything safely. And at the end of the day, it's about researchers like you guys, students who are here. What it should matter is you are safely doing this, you are safe and can go on to do many great things for India. So that should be really the focus.
Yeah. Thank you so much for such a delicate balance. So the next question from our audience, Mr. Prabhat Kumar, he's asking, is biosafety divided in subsections?
Great question. Yes, biosafety does have different subsections, and people tend to specialize in different areas. I would say it comes from primarily the different risks of working with things. So for example, animal biosafety, you can get into animal biosafety and talk about the zoonosis potential from the animal, the potential exposure when you inject the animal with what you're working with, or even more like obvious things like an animal you're working with could bite, scratch, kick you. Certain large animal models, there are labs out in Colorado that work with, say, for example, bison, right? If you need to study what pathogens are impacting a bison, a big bison, how do you keep that in the laboratory and work with it without getting crushed by a bison kicking you? So, so that itself is a whole area, and that you work with veterinarians, you work with people with specialty in that area, and you can do that. So like that, there's decontamination. There's, of course, the security aspects of it with select agents and other regulatory things. I mentioned animal work, toxins. Fungi also is kind of a unique area right now, because fungi can release spores, and it's very kind of environmentally can become an issue. That's something that is gaining more momentum. So yeah, definitely, I would recommend that you read the BMBL. It's a document. If you search just BMBL 6th edition on Google, you can see it's a big document from CDC and NIH. And also the WHO has a laboratory manual. They talk about that too. But BMBL has more sections and kind of gives you an outline of the various areas involved.
Thank you, Dr. Schwartz. Our next question from our audience is from Nabila Parveen. And the question is the world going to bioterrorism in a mannered way? If yes, then how to control safety?
So the bioterrorism question is interesting. We know that, it's known state actors. When I say state actors, like countries have focused on using biological agents, individuals have focused on doing that, but it's very difficult in terms of, there's some details that I'd rather not get into, but it's difficult to spread it without impacting yourself. So things like that have to be factored in. There are controls in place. Law enforcement agencies have specific biosafety or biosecurity units that worry about these things, right? So here, FBI sometimes ends up recruiting microbiologists. They know that kind of the background. They are able to provide feedback into what these issues might be and what to look out for. So I wouldn't say like the world is going towards that or any sort of alarming thing like that. But the more you educate the other people, the good people, they can keep an eye on what's going on, right? The more you create the awareness of biosecurity and other things, people who want to just do research for good can keep an eye on that and make sure things are not going in the wrong direction.
Yeah. So the next question from our audience is from Ms. Chala Dima. The question is, what differs lab biosafety, biosecurity from fieldwork biosafety and biosecurity?
Yeah, that's a great question. So fieldwork is another sub area, the previous question. Fieldwork, it can be a bit unpredictable and you don't have, in many cases, the engineering controls that you would have in a building. So for example, biosafety cabinet, right? If you're working with a biosafety cabinet in a laboratory, it's an engineering control. The air gets HEPA filtered before it's coming out. Your pathogens are contained inside. Working with a biosafety cabinet in the field is, you could still do it, but it's difficult, right? Because a biosafety cabinet has to be certified every time you move it, and because to make sure it's sealed properly and things like that. So as you move in the field and working with pathogens, how do you do that safely? So you start relying on PPE, and also the PPE has to function like it is designed. So all those things add additional complexities, and of course in field work depends on, are you going out there and collecting samples from migratory birds to look at influenza, for example? Are you going there and working on some wildlife species? So those dangers are there that come along with that. So it is very different, but end of the day, it all depends on the risk assessment process. So if you do a good risk assessment and put good controls in place, you can do the work safely.
Yeah, so one more question we have. The question is, how can AI and data-driven research contribute to both opportunities and risks in dual-use research?
Yeah, excellent question. That is the question that everybody is grappling with right now. So whoever asked that question, really good. What AI does, and it's not perfect, and it's not, I don't want to oversell the capabilities, but at the same time, anybody who has used AI tools can tell you the advancements we have started seeing just from earlier this year to now, how different, whether that's image generation, text understanding, reasoning, how advanced each one is getting, right? So AI can speed up certain things in terms of in biology. Let's say, the example of if you want to study how proteins fold on the surfaces of a pathogen so you can develop good therapies, the flip side of it is you can also look at what can evade those therapies, right? So, and doing that in a faster way with AI tools does increase the risk. So, but on the other side of it, in terms of having oversight, we can then, the people who want to do this, the oversight of this can use AI tools to review protocols, for example. We might be able to detect these research proposals or maybe some chatter somewhere else that AI could easily pick up that the human may not be able to, or it takes a long time for them to do, and make a lot of the biosafety tasks easier. So if I want to develop a protocol for somebody to work on, I just maybe need to do one and then say, use this as a model, now develop another protocol for this pathogen. So that in those ways it will help, in those ways it will help us, but it also has the potential to kind of speed things up and go in the wrong direction. So there's a lot of discussion now in the US and other parts of the world about how do we AI and bio. If you look up online, usually that's AI X bio. There's a lot of conversation on that, on how do we harness this, how do we safely do this so it doesn't get out of hand.
Yeah, that's indeed a delicate balance. So, ensuring progress while maintaining safety and ethics. Since science is inherently global, how can international collaboration help harmonize biosecurity policies, especially when research practices and regulations differ across countries?
Yeah, that's a great question. Again, and there are different priorities. There are different things that are important nationally for different groups. Funding is another issue. Not everybody might have the capability of even having one person dedicated for safety, right? You guys saw in my team, the picture that I shared, I have 6 staff members, of course, doing various things. But right now for biosafety specifically, we have 2.5 full-time people dedicated to that, 3.5 including me. But many institutions may not even have people to oversee those things and might not think that it is important to do that. but that's where the awareness piece comes up. You have to, during these collaborations and these proposal submission stages, it's really important to take time and say, this is how it can be done safely. Let's try to build in some funding for maybe having those oversight mechanisms. Let's try to build in some training requirements, some oversight requirements, both locations. And it's not just, doesn't have to be India, it can be anywhere in the world, right? And India could be collaborating with a neighbor in Sri Lanka, for example. And they just need to make sure to have that dialogue to make sure these safety measures are in place. Because again, I go back to this, end of the day, it could impact the worker and cause harm to the worker, and they could end up taking it home, right? If you're infected with something in the lab accidentally, you can end up taking it home and infecting your family, your community. Yeah, you don't want that to happen. So it's really important to make sure those things are discussed and things are put in place for that. Yeah.
Yeah, so that global perspective is crucial. I got a new question from our audience, so I'll read it for you. So the question is, the implementation of lab biorisk management is the roles of the top managements in a certain organization or institution as per the ISO 35001. In unknown circumstances, they are reactants to do so. How this type of issue will be improved?
Yeah, it's a good question, Chala. It's, institutions have to, so it goes both ways. People at the bench have to try to do the right thing, but if they don't have the support of leadership and safety is not prioritized, it's hard to do it, right? And let's be honest, you can only try so much until leadership just shuts it down and says, no, don't worry about it. We're not going to put any resources in there. So I think it has to be both top down and bottom up. So what I would suggest is finding champions within your leadership structure. Even if one person says like, yeah, this is important, our people matter to us, and we want to make sure our people do it safely so they can continue to grow and do great things, and our institution can be impacted by that. So if safety is prioritized, safety and security, one of the things that you as researchers, you can correct me if I'm wrong, you'll feel more comfortable going to work. You'll feel safer going to work and putting your full effort into it, right? Because your institution cares about you, your institution cares about your safety, and your mind will be more on doing the research correctly and doing big things. So I would say find champions who can get this message to others in the leadership and push this issue, because overall it'll help everybody. Yes, you may need to put some resources into it. Yes, it takes a lot of back and forth, might seem burdensome, but end of the day, it's really important to create that kind of safety and secure culture that helps you do really well.
Yeah, so the next question is, how important is training and education in shaping researchers' awareness about dual use risks, and what methods have you found most effective at Duke University?
Yeah, definitely training is important, and cannot understate how important it is. As you know, even in this conversation, there's lots of questions coming up, and once you have that awareness, then you start thinking about all the possibilities and you can ask those questions. The method that I've found helpful and I've used quite a bit is having this type of discussion rather than just, put some PowerPoint up and just say, this is what you should do and this is what you have to follow. The reason behind that is if they have questions and if we are able to clarify and make it understand why that's important, you then will not only have people who understand it and not only have people who are willing to do it because it makes sense. Now you also have created champions who will go out and convince others. They'll say, this makes sense because of XYZ, let's do it. Let's all do it, right? So having this type of discussion, being willing to answer some difficult questions, it's okay. And we learn, and also having the mindset that you don't know everything. I don't know all the processes that a laboratory does. My PhD was in a very specific area, but I don't know all the procedures. I'm open to learning that it can be done safely. I'm open to learning that maybe it's not as risky as I thought, and I can reduce my risk tolerance, towards that. So you have to come at it with different angle, and also kind of that humility to say, researchers are the experts here. We need to learn from them, ask all the questions. Both sides have to sit down and say, let's figure it out really for the greater good. But yeah, it's you cannot understate the importance of training and awareness creation, and that's why I do these things. I've given other talks in India, in various forums, because the more you create the awareness, the more people will start doing these things.
Yeah, that's inspiring. Practical education is clearly the foundation for responsible science. So, with rapid advancements in synthetic biology, CRISPR-Cas9, and AI-driven biology, what new biosecurity risks are emerging, and how should we be prepared for them?
Yeah, that's a great question. So there's also another thing in addition to all the things you listed. I'm sure students will be interested in learning about it. It's mirror life. So look that up, mirror biomolecules and mirror life. And it's a very interesting concept. I recently attended a workshop in DC and learned quite a bit myself. So all the other things you mentioned are important. Synthetic cells, for example. I personally, and again, I need to learn more about it, but if you're building a synthetic cell, hopefully you have all the right components and are putting only essential things in for the cell to work. And there could be hybrid situations, yeah, right? But typically, if you're doing from scratch, you would know exactly what you're putting in. So we could do risk assessment of those components and say, okay, is this safe or not? Similarly, really depends on the application side of things. Some of the issues that are emerging, like mirror life, mirror biomolecules, what it does is it's different than the natural form, and its chirality is different to explain. It's different. It's kind of like a mirror form of it. So then detection becomes hard. Potentially decontamination becomes hard, and other things, become hard. So these are issues that could happen in the future, and also on field tests and things like that. When you do gene drive experiments, how does one thing, if you're trying to say control mosquito population using gene drive, how does that impact the environment? What does that change just because you've altered that? It's a nuisance, but what things are dependent on that mosquito population? How is that affecting the entire ecosystem? So these are things that I would say are, and there's much more, I would say are things that we need to collectively think about and try to address.
Yeah, indeed. Technological innovation brings both opportunity and responsibility. So, finally, what advice would you give to students and early career researchers to ensure their work contributes positively to science while minimizing risks of dual use?
Yeah, so I mean, specifically to students and early career people, it's, as I said earlier, just learn as much as you can about this. And you guys are doing a great job by doing more than what's required, right? I mean, just organizing this webinar series, bringing in different perspectives. You're already in the right path and trying to learn more than what's required, and continue that in a initiative that you're putting in, so that you can be well informed and make good decisions, and have an open mind in terms of the actual risk, and don't go in with like this is it. Be willing to learn and change your opinion based on data. You could have a very strong opinion and very strong conviction, but if you see data that kind of contradicts that, be willing to change that and kind of evolve with that process, right? I think that'll be helpful for everybody if you're willing to do that. And then overall, just I would say, talk to anybody who would listen, right, about these issues, and create that awareness, and go out there and kind of create more of that awareness. So there's statewide dialogue, national dialogue about this, international dialogue. And as I said in one of my slides, India has the brain power and has the expertise to lead these conversations in the global south or in the world. So really start there, and it starts small. And you guys can someday be all those people who do those type of things for India.
Yeah. Thank you, Dr. Schwartz. I wish we could continue longer, but our time is limited. Esteemed guests and fellow students, this has indeed been a remarkable session. I take this opportunity to extend my heartfelt thanks to Dr. Anthony Schwartz for his valuable time and inspiring insights. I would also like to thank all participants for their active involvement during this session. Now, I hand it back to our MC, Ms. Alicia. Over to you. Thank you.
Thank you so much, Ms. Fatima, for expertly moderating the session. As we conclude today's session, I now invite Ms. Ikra Bashir to deliver the vote of thanks on behalf of the organizing committee. Ms. Ikra, over to you.
Thank you, Alicia. Good evening, everyone. It is indeed an honor and privilege to deliver the vote of thanks on behalf of the organizing team and the Center for Scientific Research and Development, People's University, Bhopal, India. As we draw the curtain on today's enriching session of the BioConnect webinar series, we are truly grateful to our distinguished speaker, Dr. Anthony Schwartz, for his valuable time and insightful talk on dual-use research and biosecurity, building a culture of responsibility. Sir, your thoughts have enlightened us and provided meaningful insights into dual-use research and biosecurity, helping to build a culture of responsibility. We express our deep gratitude to our Honorable Registrar of People's University, Dr. Neerja Malik, for her presence. Ma'am, your encouragement and continuous support serve as a great source of inspiration for all of us. We extend our sincere gratitude to our mentor and guide, Professor Dr. Subhash J. Ghore, Head of the Center for Scientific Research and Development. Sir, your vision, guidance, and constant support have been the driving force behind this webinar series. Your ensuring encouragement has been the backbone of today's successful session. Heartfelt thanks for our moderator, Ms. Fatima, for conducting the session with clarity, confidence, and depth. Your thoughtful questions brought out the best insights from our speakers. We are thankful to our chairperson, Ms. Aisha, for her support. Her efforts are appreciated. Before we close, I would like to thank each one of you present here today. Finally, we want to record our special thanks and most profound appreciation to the management of People's University for their unwavering support and encouragement. The support of management for student-led activities such as the BioConnect webinar series enables us to dream big and achieve more. With that, I hand it over to our MC to continue with the proceedings. Ms. Alicia, over to you. Thank you.
Thank you so much, Ms. Ikra, for those words of appreciation. Once again, thank you to everyone who joined us today. We encourage you to share your thoughts on the event. The post-webinar survey link is now posted in the chat box. Your feedback is really very important to us. Please complete the feedback form using the link posted in the chat box. Now we will wait 2 to 3 minutes for you to complete the feedback form. We really hope you have completed the feedback form. Please note that your e-certificates will be sent to you shortly. We look forward to welcoming you to our next BioConnect webinar session very soon. This is Alicia Mansouri signing off on behalf of the Center for Scientific Research and Development student team at People's University. Have a wonderful evening, everyone. Goodbye.